recent results. Okay, got it. About some recent results with them in my group in my institute, also with other colleagues in CNR and outside. And essentially, I'd like to present the organs on chips, some experiments made by biophysicists and biologists around these devices. And a first attempt to build up a macroscopic mathematical model, some simulation, and also calibration of the model and particle visualizations, how to come back from cells to density, so come back to particles. And finally, some few, if I have time, some few remarks about a microscopic model we build up to as an interstitial way between the experiments and the microscopic model. So these works are in collaboration essentially for the experiments biological data with people in the Institute of Photonics in Rome, Francesca Romana Bertani, Luca Businara, Dell'Edinino, and Maria Gerardino. In my group, essentially Gabriela Bretti collaborates in all these works, and we have PhD students now finishing Rome 3, Alicia Brown, who has now a grant with the Institute. For the data analysis, we had some support from Davide Verni and Paola Stolfi, and this recent part about microscopic models, that's still in collaboration with Gabriela Bretti and Nicola Roselli, who is a PhD student in Sapienza and also affiliated to the Institute. Just to say, all the collaborators say the following, I will skip the contributions, the owner of the contributions. Organs on chips are quite recent devices, 15 years old, something like that. They try to reproduce human organs, and the idea is to test drugs and really in a more realistic context, in the sense that usually you have a Petri discs and then animals and humans. And in the middle, you can have this kind of devices where you try to mimic, I don't know, lungs, brain, heart, and so on. And you try to apply drugs in this kind of micro environment. Here, for instance, you have some chambers where you put cells of different types, spleen cells, melanoma, and so on. And you have channels joining these different chambers. And so you can administrate the drugs or just look with a video time lapse and record what's happening. And here is the typical situation we are going to study today. Essentially, we have this kind of chip, and we cut just one rectangle where there are some channels joining to different chambers, where we have in our setting essentially immune cells on one side, cancer cells on the other side. Here is, for instance, an example. We have two kinds of cancer cells. Essentially, we worked on knockout cells, which are changed to be inactive, so inactivated, so to see just the effect of the response of the immune cells. And here you have wild type cells moving around the two chambers, passing inside of the channels and so on. Our, just to fix the ideas for you, look at this movie. I hope it's you can see. On the right-hand side, you have the small particles like snowflakes moving are immune cells. On the left, we have the glial cells, which are knockout cancer cells. And the immune cells pass in the channels because they are attracted by some chemoattractant released by cancer cells, and they try to attack the cancer cells to eliminate the cancer cells. And so that's what we have in mind when we try to understand this phenomena, tracking the movement of the cells and trying to build up a model to reproduce this kind of experiments and to extract the relevant data to try to understand the phenomena. What are the factors playing some role in this kind of experiments? So our first point is derivation mathematical model to describe this kind of phenomena. And we take the position of the cells. Thanks to some tracking algorithms, we know the trajectories of the single cells. And so we can use this data to fit our mathematical model and to try to have a robust calibration of this model. And so we want to, in part, our first attempt is to translate the data, the particle data in a density. And that's a problem because we are a discrete set of data and we want to compare with the density field. So we have to, in some sense, to translate to one reality in the other. So what's the idea in this, considering in this organs on chip? We have the experiments. We try to build up a model, and thanks to the experiment, we try to have a confirmation. We can have new results and prediction, and so we can back maybe suggesting new results, new experiments, and also trying to connect this organs on chip community to the real physiology. I mean, here we have a few thousand of cells, but in the reality we have billions of cells. So a macroscopic model can be meaningful in connecting these different scales. So first of all, the first effect we have to understand is the chemotaxis. So we are going to use standard models in chemotaxy. I recall, but maybe you know very well that it's just chemotaxis is the release of a chemoattractant and the cells at different level, amoeba or in angiogenesis, so fibroblasts, stem cells, and so on. They are moving according to gradients or even concentration of some chemical component. And the standard model for that is Keller-Segal model, which is now quite old, 50 years old, 51 years old, and is essentially composed by two equations. One equation for the density of bacteria, cells, individuals, which change the concentration in space by a diffusion term and a transport term given by a chemical gradient, and another equation, which is usually a diffusion equation for the chemoattractant or chemorepellent sometimes. And we have a source term where we have an interaction between the cells and the chemoattractant itself. Sometimes you have a production or even depletion of the chemoattractant. It exists a large theory for this kind of models. So in 1D, there are global solution. In 2D, there are existence for a family of these models for a small initial data, blow up for greater solution. But we are not concerned with this kind of problems because it's possible to change the equation to avoid blow up of solution into dimension. We are going to use also an hyperbolic alternative, the Greenberg-Alt model, which is essentially a kinetic model. We have two families, the cells going to the left, cells going to the right, like Goldstein-Taylor. But here, the turning rate is just, influenced by some chemoattractant, which is solving the same equation like before. If you rearrange the components, and so you take the total density of cells, so U plus U minus, and you introduce a total flux, so gamma which is the speed of the cells, which is assumed constant, u plus minus u minus, so the total flux of cells, you obtain a Cattaneo model. A Cattaneo model where here the coefficients are influenced by the chemoattractant. So essentially, they are very similar, this model, the Keller-Siegel and this one. If you just skip the time derivative of V in the second equation here, you obtain the same model as before by standard procedure. So, I mean, they are connected. This one is a finite, we have a finite speed propagation, and the other one, you have an infinite speed, which is not really physical. Another extension we are going to use is to put this kind of equations, and now the hyperbolic, but for the parabolic ones is even more standard, on a network, because we want to put in connection the chambers and the channels in some sense. And I anticipate this idea that the channels are one-dimensional in our model, and the chamber are two-dimensional. So we have to connect the domains in a different dimension. And if you take the equation on a network, you have to give some boundary, essentially transmission conditional node. For the parabolic equation, you can use a Karen-Kachowski transmission condition, which are proportional. So the flux is the same on the right and the left of each node, and this flux is proportional to the jump between the density on the right and the left. So you have no continuity at node, but you have a jump, because maybe you can imagine you have a membrane, you have something on top of the membrane, and you have some liquid passing on the other side. And so it's natural condition. For the hyperbolic case, you can do the same, more or less. And we started with a single equation treating traffic flows. And we introduced a new family of transmission conditions, which are the analogous of Karen-Kachowski in the hyperbolic case. You can look at that just looking at the dissipativity condition. You want to dissipate energy at the node. One possibility, first you have to have the continuity of flux, but also you want that the energy is dissipated. So you have that V1, the flux, which is the same on both sides of the equation, times the jump of the density is positive. And you can choose or to have the same density at both sides, like for instance, in elasticity problems, shallow water and so on, which is essentially energy conservation. Or you can mimic the Karen-Kachowski parabolic condition, and you can have a jump like in traffic. In traffic, when you have a bottleneck, on one side you have a lot of cars, on the other side you have very few cars passing. So it's natural to have different density. And for the cells, we expect to have the same effect. So it's our choice in this case. And we elaborated general theory with general condition. Now I skip this part for many junction parabolic, hyperbolic system, and we have a general theory. And with Francesco Guagliini, we established a global existence for this kind of models. And with Gabriela Bretti and Magali Ribot, we implemented some numerical algorithms, and even in more complex networks, in MAIDs and so on. And also
Excuse me, may I ask a question? Because in traffic flow, to avoid some kind of singularities, I'm not an expert, but sometimes they introduce buffering, or which should model a roundabout or something like that. There is no need to hear or something like that?
Not really, because you have conservation of mass. So, I mean, when you enter a channel, the cell is entering a channel, or sometimes, for instance, in the artificial scaffold, you have also real wires. And I mean, you have no buffering on this part. You know, the mass is conserved anyway. And for buffering is more used for internet modeling. and in this case, or a supply chain or something like that.
Okay.
But not in this case.
Not in this case. Okay. Good.
So now I present our model with this framework in mind. So what we do is we have 2 chambers, as in the movie, and we have a lot of wires because they are one-dimensional, connecting one chamber with the other. So in the right chamber, we are going to put the immune cells, and in the left chamber, we have to put the cancer cells. And it's possible to look what happens. The equations are tumor cells are T, M is for macrophages, so immune cells. And the model is in the chamber is 2-dimensional. So you have the standard diffusion model for the cancer cells, and for the knockout, essentially this term is the diffusion term is equal to 0 because they are stuck on the field. You have a mortality given also by the influence of immune cells, which are going to release some chemical enzyme to degrade tumor cells, and also there is some natural environmental decay of tumor cells. You have another equation for the immune cells, which is diffusion and chemotaxis. And here, okay, I skip one term. The chemotaxis is given by phi, which is essentially the chemoattractant released by the tumor cells. So, and also there is some decay of the immune cells, which is neglected in the following. And the chemoattractant is so diffusing, produced by the tumor cell, and we have a degradation of the chemoattractant during the time by enzyme in the environment. And also you have this other chemical produced by the immune cells to attack the cancer cells. So this basic model, very simple, where we have a lot of constant, we don't know the values of this constant. Sometimes in the literature we can find the value of this constant, but it's very difficult to assess experimentally the real value. And in particular,
excuse me, Roberto. I mean, in some models, the chemoattractant is released by the macrophages themselves.
Yeah, but not in this model, so because here the attraction is produced by the tumor cells. Okay, there are some internal mechanisms of signaling between immune cells, but here they are completely neglected. The real effect is due to the presence of tumor cells, which are releasing some chemicals, which is what produce the movement of immune cells. So without the tumor cells, they don't move. Essentially, they have a small random movement around, but they have no precise direction. So the internal, so that's different from classical model, because you have this coupling, which is a sort of action, mediated action by the chemoattractant released by the tumor cells. Okay. And you have the equivalent hyperbolic model, which is a one-dimensional, it's in the channel. And it is one-dimensional because essentially for 2 reasons. So the first one is in the channel, we have a very different mechanism of transportation, which is a constant speed. They move with a sort of, they swim inside of the channels, but really you have no diffusion at all. And so it's realistic to think about this kind of Greenberg-Out model, but also because we could apply this model in 2 dimensions, but be careful because the Cattaneo model in 2 dimensions is ill-posed because we have as well known negative solution in finite time. So it's otherwise we could apply the hyperbolic model everywhere, but we prefer to use this kind of different model for different dimensions. The chemoattractive function, which is very important, which is what we put in front of here. Okay, I skip the phi. The key function, which is the strength of the gradient is maybe constant, or you have a saturation when the receptor are there is too much density of gamma attractant, or also you can avoid the overcrowding. So, for instance, the macrophage, you can avoid blow up just putting a sort of behavior, which is you have a maximal value of macrophage, so you cannot there is no blow up occurring. But in this kind of model, we have a very small density, so in general we have no blow up at all. So this term is not really useful. And for each chamber, we connect using essentially just I put as an example the Kedem-Kochalski, so the flux, which here it is also with the gamma attractant part. It is not just a gradient. Is proportional to the difference of density of cells on one side and the other. And that's the way to connect also the different dimensions, so domains in 1D and 2D. So, I mean, it's a delicate part, essentially from numerical point of view, is quite delicate to try to implement this kind of models. So on the external boundaries, we have no flux conditions, which is not correct, because you have seen at the beginning, it's just a rectangle cut off from a more general problem. But okay, for just to assess the validity of this kind of approach, we can imagine at the beginning to have a Neumann condition, and more general condition can be implemented quite easily. We use, I skip the details, Crank-Nicholson models, methods for the diffusion part, and asymptotic high order schemes we developed with Magali Ribot and Gabriela Bretti for the hyperbolic part. And so we have conservation of mass on the nodes. Here you have just a quite schematic version. T is tumor, M macrophage, Phi is the gamma attractant, and W is the chemical produced by immune cells. At the beginning, you have zero chemicals. Tumor cells are concentrated on the left, and here on the right, we have just the yellow strip where you have just macrophage, and that's a time equal to 0. And that's what we want to look is because tumor cells essentially are not really important in this kind of experiment, but a macrophage can move differently according to the different chemotactic sensitivity we put. For instance, in the basic model, we have this kind of expansion. If there is a receptor saturation, we have more accumulation on this side, and in the overcrowding, we have an intermediate result. So, I mean, we can implement this kind of model, and we want to compare this approach with the data. So the problem is that we have this observation. We track the cells. The blue one are the cancer cells. The red one are the immune cells. We have the tracking all the time at 2 minutes frame. And we can mark up, for instance, you have the big ball, white ball, automatic markup of the immune cells. And so the first point is to transform this information, which is pointwise, in a density. So there are many different kind of methods to translate in density. But the first one, which is you take a grid, you count for each grid the quantity for each square, the number of cells that you find at each time is a very, unstable, because if you change the grid, you change the number of cells, so you have for very quite bothering effects given by the kind of grid that you have chosen. A standard method is to take some kernel Gaussian kernels, and to use to take the sum, to take the average of the Gaussian kernels, and so you just sum up the Gaussian for each cell you have in your video, and you obtain a 2D density estimation of the cell, so which is robust for our purposes. Clearly, you have to take the amplitude and also the variation of the kernel density in a right way to avoid oscillations or too much smoothing. And here, that's some rule of thumbs to obtain this kind of model. Here, there are some technicalities, some multiplication for some metrics to have different effects, and it is possible to see the effect of this transformation according to the metrics you have chosen inside of the kernel estimates to take the results. But anyway, that's quite well established in the community of people using data, pointwise data to transform in density. It's a quite well-known technique. And you obtain something like that. You take the data, and you can translate in a density. Here you see the accumulation and the cells passing from one side to the other. This one is just a reconstruction made by using kernel density estimates for our original experiment. So what happens when we compare our model with the data? We recall we have all these models in 2D and 1D in the channels and so on. You have a lot of parameters which are completely free. It's very difficult to reconstruct. So we take a simplification. We have to minimize a functional on the data. And this functional essentially is made by using some Tikhonov regularization. So you have the density given by the data, your density given for a given set of a parameter, and you add some regularizing Tikhonov terms, which are L2 norm of some function g or h and so on, which depends on the data, or h1 function or L2 function of the density, just to stabilize your approximation, because as you know, this kind of inverse problem are unstable. And so essentially what we take is least square functional, and what we take a matrix which is a sort of a Lagrangian multiplier here, the regularization parameter, and here you have a matrix which is chosen to try to avoid a lot of instability in the minimization part. And so according to, for instance, here is in 1D, you have no regularized, you have a lot of corners, and the exact solution is the black one, and you have a good approximation by using this kind of minimal minimization with Tikhonov techniques. So we have another problem now that usually you have a lot of local minima, and the local minima are too much to be chosen in a meaningful way. And here another technique is to use a coarser mesh grid. So when you have a larger grid, you have less minima, and so you can try to focus on this kind of minima, and then you can perform your minimization around this local minima. Here is an example for a standard transport diffusion equation. When your grid is 1 over 100, you have a lot of minima here. And you reduce when you have just 1/2 size. So you have a very rough grid, and you have just local minima, and you can choose what kind of minima you have to look off. And when you apply this kind of multi-grid techniques, you can reconstruct for a given speed and a given diffusion. You take your data, you translate in something which is with a Gaussian noise as a sort of experimental data, and you try to recover your original velocity field and the diffusion term, and using a fixed grid, you have a very large error, especially for the transportation part, which is reduced when you use this kind of multi-grid with a coarser grid method. So it's possible, okay, to have a good estimate of what you are doing. And here is for a fixed V. You can imagine the difficulty to when a V is depending on the space and time, clearly. So we simplify a lot our original experiments. So we take just a sub-region. We skip off the channels, which are in some sense just used to have a global, the reproduction of global experiments, not for the calibration. We skip the cytokine, which is produced by the immune cells, because in this time frame, which they are not really important. And also the tumor cells are completely stuck. And so at the beginning, we have two equations, but we have no data for the chemoattractant, because in the experiments we have just the position of the cells. That's a big, quite a big problem, and we tried with different group of biologists, but that's very difficult to give a measurement of this chemoattractant. So you have this equation for the macrophage and the chemoattractant given by a fixed number of tumor cells. And we can use the first equation. We can recover V and the diffusion of the immune cells, and then by another minimization, we can try to recover phi by another minimization procedure, using the fact that phi is a solution of diffusion equation. Without using this important information, it's impossible to close the gap between the two equations. There are too much parameter and not enough information to do that. But using this second information by restricting our attention to the gradient of the solution of the diffusion equation, it's possible to have a quite stable result. And so what do we do at the end of this story? We start from the experiments, we track the cells, and so we have this trajectories of the cells inside of the chambers. We fit the model, we replicate, and we come back. We can, from the density, come back to the particles. And our final representation, which is useful for biologists, for instance, is again in terms of particle, which are the particle given a density, we can extract particles. So you see, on the left, we have the experiments. On the right, we have what we have created artificially by using our model, which is very similar, even if we have a quite, quite different regime. We have a different densities of cells because there is an error, but it's acceptable, and in some sense you can reproduce the reality. So this approach is useful, is interesting for people working with this kind of device, but we have still a large jump between our data on particle and the density. So our idea, just in these 3 minutes, 1 minute maybe, I'd like to say that it's possible to, in the middle, to elaborate some Newtonian model for the cells. So individual based model where we have essentially a Newton equation where chemotaxis, the action of cancer cells and immune cells, attraction, repulsion, etc., are taken into account. The chemoattractant equation is the same. And so we have an hybrid approach, which is in the middle between experiments, our macroscopic model. And we have these recent results. We obtained just a preprint some weeks ago. We just cut up of our experiments a few cells, 4 cells, tumor cells, and we try to reproduce by this kind of hybrid equation. So chemicals are still in the partial differential equation, and the immune cells are ordinary differential equation, and also using some stochastic perturbation is possible to reproduce quite well the statistics of this kind of cells. So in the calibration is not trivial. We have to reconstruct by interpolation the velocity field of the equation from the experiments and to compare to the single cells we are going to use. And the real goal of this model is to start some experiments, maybe improve our microscopic model using the microscopic one, and extrapolate to the real, the real organs, a full liver or a full lung in the future. Here, a small selection of some papers which are of interest for this kind of research. And I thank you for your attention.
Okay, thanks to Professor Natalini for his very interesting talk. There are some questions.
If I have I have a question. I mean, okay. With this calibration, I mean, you mentioned that it's a big. I think the big problem is to understand what's the right form of the chemotaxis.
Yeah.
Saturation effects and whatever. Yeah. What is the most appropriate? Because there is a large degree of arbitrarity. I mean.
Yeah. Up to now, the saturation one is working quite well. Mm-hmm. Because in fact, from a biological point of view, you have that the receptor, mm-hmm. They start to work. You have a lot of receptors on the cells. When these receptors are full, they stop to be activated. So, mm-hmm. You have a loss of activity. So your chemotaxis is no more working. So it's quite natural to use the saturation one. The exact value of the exponent in the saturation is a matter of debate, but using this kind of approach by inverse problems, essentially Tikhonov regularization, we have a quite good stability in our results. But clearly, we have not enough data to predict the general situation. Here, what we know, and I didn't say during the conference that we have tumor cells on one side, but outside of this rectangle, we have a sort of a constant gradient, which is linear, more or less, given by a well, which is just outside of our view. Mm-hmm. And so we knew that essentially we have a small variation around this constant slope. And so we have a good starting point. So in some sense, we were cheating.
Okay.
I have also a question, if I can.
Please.
May I?
Yes, please, Maria Carmelo.
Yes, Roberto. So in one of the last slides, you showed us the comparison between the experiment and the simulation with the
this one.
Yes, here. So if we so I think that A should be compared with B. Is that correct? And C with D. Yeah. So what I notice is that this sort of accumulation of red cells on the.
That's a problem with the model. Yeah, I mean that's a very rough calibration, and we know.
So that's the question: is if you have an explanation for this qualitative difference.
Yeah, I think that the real problem is that probably there is too much accumulation of chemotaxis on this region, on this part, in the model, in the mathematical model, which is not really present in the reality. So at least here, at the entrance of the channel on this side, you have this accumulation. So I mean. There's a problem that we calibrate on a small portion of the right chamber. And so what happens on the left chamber maybe is not really accurate.
Yeah, maybe most of the calibration is on the tumor cells.
No, because the tumor cells are stuck, because this one is the knockout type, which is essentially they are stuck, they have some drug, and they are just producing this chemoattractant, they are doing nothing. And the big problem is this calibration of the chemoattractant, which is unknown, because we have no measures. So that's the problem. And I agree that's the big problem in biology. We asked it to a lot of people working in this field with different laboratories. Even in Paris, I discussed, we have a joint PhD thesis with a group of Benoit Borstam on these kind of problems. And we have no measurement of this.
But the red cells, with the red cells, you indicate the chemoattractant, right?
No, the red cells are the immune cells.
The immune cells.
You don't see the chemoattractant, but you deduce that there is chemoattractant because they are all in this part.
Yes, they are. Yeah, okay. Okay, thank you.
Okay,
you're welcome.
Other questions?
Yes, if I may.
Yeah, Andrea.
Thank you. Ciao Roberto, ciao Marco, ciao Maria Carmela. Let me back to English.
Oh.
First of all, thank you very much. Very nice presentation. Very attractive, if you will. Pardon the pun. Two questions. The first is, you decided to use a two-dimensional model on the left chamber and on the right chamber, and a one-dimensional model in the connecting channels, what they are. Fine. Now, I understand this has a reason, because these cells essentially move in a straight line from one component to the other. But I wonder whether the complication is worth the effort. Couldn't you just throw in a single two-dimensional model throughout and be done with it?
Not really, because I mean, these channels, in some sense, mimic a membrane. Okay, the idea behind these channels is to create a sort of membrane, which is what usually cells have to cross to reach cancer cells and so on. Okay, so the idea was to have a physical obstacle. Okay, and if you put all in two dimension, you have to understand that the channel, the diameter of these channels is the same of the cells. So really, they cannot move in the vertical direction. So, okay, another possibility is just to skip out these channels, because also we have no data. And so eliminate and just to look at what happens. But then you are in trouble, because you have one cell enter on one side, and the other you have another cell exiting on the other side, and you have no connection. Thanks to this modeling, we can take track of what happens inside. And even if we have a few data, our model is quite realistic. So, I mean, the problem here is more with the chemoattractant, not with the movement of cells. A two-dimensional model of the channel is not the right one, because, I mean, really, you could put a 1D diffusion model in the channel, maybe, but still you have a too fast propagation. You have a fast propagation of the cells inside the channel if you put a parabolic model. So this transportation, this kinetic model is the more adapted. We tried anyway to try to have the best result, which is the result more similar to the experiment. And so because there is a time which is not very long of permanence inside of the channel, so you cannot forget the existence of the channels.
Okay. The second question is this. You mentioned the eventual goal of this modeling effort is to actually give biologists some information about what happens in the microscale in real organs, like diffusion of whatever. Yeah. So parameter calibration seems to be an important part or important component of this thing. For instance, when you are looking at the sensitivity of these immune cells to chemoattractant gradient, and that is a parameter in your model, of course, this it cannot be distinguished from the actual gradient if you don't know it. So you have two parameters, let's say, one related to the formation of the gradient and one related to the sensitivity of the cells to the gradient. And these two parameters are intrinsically indistinguishable.
Yeah, you're right. But I mean, but they have a different effect on the trajectories of the cells. So what we are using is using the movement of the cells to deduce. I mean, since we separate the two effects. I mean, we have a vector field, which is the multiplication of chemosensitivity and the gradient, and then we say, okay, assume the chemosensitivity is made in this form, 1 over 1 plus phi power gamma, for instance.
Whatever.
What happens if phi is the solution of this equation, and you find some consistent convergence of your calibration? So, I mean, I agree with you. That's the delicate point, and maybe the more delicate point of this stuff. The fact that we try to reconstruct the chemoattractant just from the trajectories of the cells, but there is no other solution up to now. I mean, in my opinion, even the relevant results is to try to give some new information, but clearly we are just at the beginning of this kind of story.
Okay, thanks again.
No, just I wanted to make a, I mean, it was the first question of Andrea was...
Another question.
Yeah, if I'm allowed.
Okay.
Well, you probably mentioned that. Why not, did you think to put a porous medium equation so that there's a slow diffusion? Just...
You're right. Oh, you're right. Okay. In particular, in my dreams, the best equation is, in fact, the Preziosi model, which is, I mean, nonlinear, just like pieces. I mean, isentropic gas dynamics. And so, in some sense, with the nonlinear pressure law, which is similar to porous media equation in the diffusion limit. Okay. But the problem is that, okay, so in principle, yes, you're right. We started simple. So my answer is, first of all, maybe porous media equation is okay, but my experience with the comparison between the Preziosi model, which is Euler, and porous media, I prefer the hyperbolic one. The problem is that the relevant mathematical relation to solve these channel problems with the Preziosi model is a work in progress I'm doing with Magali Ribot, and it's quite difficult. And so we have no technology to use this kind of model in the hyperbolic setting. And the porous media has some defect that you have some artificial aggregation. In particular, with the Preziosi model, you have some bump organization of cells in some consistent way, which is what we expect. In the parabolic model, we have essentially a movement of bumps towards the boundaries, which is not well understood. And so I'm not really confident that the porous media approximation is good.
Will be better. Okay, thanks.
Okay, thanks again. Now we stop 5 minutes or not?
Maybe yes, we resume at 4.
I think it's better.
5 minutes break.
Okay, now at 4 o'clock again. Okay, next speaker is Professor Laurent Desvillettes from Institut de Mathématiques de Joseph Paris, Rive Gauche. And this talk is about the entropy structure for cross-diffusion equations. Please.
Okay, thanks a lot for the invitation. I'm very glad to speak here online. Of course, I would have liked to come to Palermo, but I hope it will be for the near future. Anyway, so what I will present today is a few ideas about the entropy structure of cross-diffusion equations. And it's a theme on which I've been working for many years now. And here is a list of the people with whom I've been collaborating on the so-called non-triangular case. I will explain this a little bit later. So there was Thomas Lepoutre, Ayman Moussa, and Ariane Trescazes and Helge Dietert in France, and also Esther Daos and Ansgar Jungel in Austria. So let me present briefly the so-called Shikesada-Kawasaki-Teramoto model, which is usually called SKT in the community. So this model is devised for understanding segregation between two competing species. So you imagine that you have, let's say, two species of animals, and you look at their densities, which are called respectively U1 and U2. And first, you take into account the birth and death of the individuals, and this is more or less the right-hand side of the system. The usual assumption is that this can be modeled by a Lotka-Volterra type, let's say, term, like this R1 minus S11 U1 minus N12 U2 times U1. So in which you have a part which is linear and which is growing, which represents in some sense the reproduction of the individuals. And then you have this negative quadratic part, which represents the competition, the effect of the competition on the evolution of the population. The S11 and S22 is called sometimes the intra-specific competition, which means that there is competition between individuals of the same species, while the S12 and S21 is the so-called interspecific competition, which means that there is competition between the individuals of the two different species. So this part is quite classical, and you can find that in textbooks like, for example, the book by Murray. It's very well explained. And there is already a lot of things to say about the system of ODEs, but it becomes much more interesting if you add to that the spatial structure, and you're looking at the, let's say, the way the individuals are moving around. So in first approximation, you imagine that they will follow some diffusion, and so the most standard equation would consist, when you take into account the space, to add this term, which is Laplacian of D1 times U1 for the first one, and Laplacian of D2 times U2 for the second one, where D1 and D2 are diffusion rates, which are typical of the species you're looking at. However, if you look at models like this, it's very easy to see that you cannot have any type of Turing instability. And because of that, it's not possible to see, in some sense, the segregation which can be seen in nature just because of the competition which is in the right-hand side. And this is the reason why those three people, Shikesada, Kawasaki, and Teramoto, they devised this model in which you add in the diffusion two new terms, these so-called A12 U1 U2 in the first equation and A21 U2 U1 in the second one, because they discovered that when A12 and or A21 are big enough, then you can produce Turing instability or Turing style instability in the model, and you end up with steady states which are stable and in which you can see the segregation, which means in some sense that you have regions in which U1 is dominant and regions in which U2 is dominant. But it can also, I mean, these equations can also be thought in terms of modeling, and they are reasonable in terms of modeling if you think that the species you are considering have some kind of intelligence, and they try in some sense to avoid each individual of the species one, let's say, will try to avoid the individual of species two. And the way this is done is that if one individual of species one sees many individuals of species two around him, then he will increase his diffusion rate. So at a microscopic level, this would mean that he would try to move away very fast from the other individuals. And at the macroscopic scale, this just means that you have a dependence of the diffusion rate for the species one, which is increasing with the concentration of the species two. And this is what you can see in the equation. The diffusion rate of species one here is something which is an increasing function of the quantity U2. And the same for the species two with respect to the species one. Okay, so this is the basics, let's say, of the modeling of this model. Now, those terms in which the diffusion rate depends on the other concentrations, they are usually called cross-diffusion terms. And well, typically in those models, we add the Neumann boundary condition, and we assume that all parameters are strictly positive in the so-called non-triangular case. The triangular case would correspond to the case, for example, when A21 is equal to 0, but not A12. And then the mathematics is quite different. I will say a few words about that at the end of the talk. So for the almost all the rest of the talk, I will focus on the case when all the parameters which appear in this equation are strictly positive. When you try to do the mathematical analysis of this system, you are very quickly blocked. And basically, the first thing that you can do is to take the total mass of the concentration U1 and of the concentration U2 and try to see its evolution. So you just integrate the equation on the wall space. This makes the Laplacian term disappear because of the Neumann boundary condition. And you end up with the two equalities which are written in the middle of the slide, in which you see that the integral of U1, I mean, its derivative in time, is proportional to itself, minus terms which are basically quadratic. And so because of this, if you now put all the non-negative terms on the same side of the system, you get an estimate which tells you that U1 and U2 are basically in L infinity in time with value in L1 in space, and they are also in L2 of both time and space. And once you have done that, in fact, it's very difficult to obtain something else, some other a priori estimate from the equations. And people were blocked for a lot of time with that. And in some sense, up to the mid 2000s, there was no progress on the general case, let's say, and people focus on special cases. Either they were considering small time solutions, or they looked at dimension one, in which you can do a little more estimates, or they suppose that some of the coefficients were equal, like the diffusion coefficients, and then you can add and subtract the equations and things like that. But there was a big improvement in the theory when, I think it was in around 2005, Li Chen and Ansgar Jungel discovered that in fact there is a hidden Lyapunov style functional for the system. And most probably they were able to see that because they were specialists of let's say entropy method for kinetic equations. And in those equations, the entropy which naturally appears is this quantity, which is u log u, or if you prefer u log u minus u plus 1, if you like positive and convex quantities. And so they probably tried to see if this gave an entropy, and actually the part of the computation which is far from being obvious comes from the fact that in order to get a Lyapunov functional, you have to put the right coefficients in front of the entropy for U1 and the entropy for U2. And those coefficients are those A21 and A12, which appeared in the equations in the upper part of the slides. That is the terms which are in the cross diffusion, which are really forming the cross diffusion. So somehow there was this big progress when they discovered that you had this functional, which was in fact not really decreasing along the flow, but not so far. And if you really write the estimate, you end up with what is written now on the slide. That is the evolution of this functional plus terms which are positive. Actually, it's a plus and not a minus in front of the A12. Sorry for that. So this quantity plus a certain number of positive terms is bounded. And typically, if you integrate that in time, you end up with a natural bound, which is that the gradient of the square root of the density is bounded in L2. Or if you prefer, the square root of the density is bounded in H1 in space. Now, this is really key in studying the problem, because once you know that, in some sense, it means that you have some compactness in the problem, because you see that you can control some gradient in X. And then you have an equation which is of parabolic type. So you can use Aubin-Lyon's lemma or some variant of it and end up with a sequence of approximating solutions from which you can then extract a subsequent which converges almost everywhere towards something which happens to be a weak solution. It's well, what I just said here in one sentence. In fact, it needs like maybe 20 pages to write down very precisely, because there are a lot of difficulties with the approximation in this problem. But anyway, the really the core of the proof is that you have this estimate which is written here. So that was really a very big improvement. And so after that, maybe one of the main questions was that is it some kind of miracle that just for this system there is this functional which is almost a Lyapunov functional, or is there some hidden structure which justifies in some sense that for many more systems in which you change, for example, the exponents and instead of taking quadratic, you take a little different, does it still work? And so close now to 10 years ago, a little less than 10 years ago, together with Thomas Lepoutre, Ayman Moussa, and Ariane Trescazes, we're able to see that actually you can change almost all the exponents in the equation. So typically in the reaction part, instead of taking S11U1, you can take S11U1 to some power, and you can do the same for the other terms. And more importantly, in the cross diffusion part, you can replace the A12U2 by A12U2 to some power, which is called here alpha 12, and the same in the second equation. And actually, you can still find a Lyapunov functional or an almost Lyapunov functional in this case, provided that alpha 12 times alpha 21 is strictly less than 1. So there is actually some kind of condition on the strength of the cross diffusion, under which it's still possible to have a Lyapunov functional. Now, strangely, this does not really include the previous case, the case of the original SKT model, in which you had both alpha 1, 2 and alpha 2, 1, which were equal to 1. And in fact, we still don't know if it's possible to do the computation if you have alpha 1, 2 times alpha 2, 1, which is equal to 1, but when alpha 1, 2 and alpha 2, 1 are not equal to 1, both of them. Well, anyway, so in this sort of generalized case, the idea is to move away from the U log U type entropy and to replace it by a power-like entropy. So this is sometimes called the Rényi entropy in the framework of information theory. Basically, so you have the functional which is written at the end of the slide, but what you have to just to keep in mind is that you have inside this term which is uj to the alpha ij, which is a power, in fact. And when you do the computation, then you find an estimate which is written just above, which looks a lot like the estimate which was appearing in the original SKT system and which was discovered by Li Chen and Ansgar Jungel. So instead of having gradients of square root, you have gradient of square root of some power of the uj. And provided that alpha 1, 2 and alpha 2, 1 are strictly less than 1, this still works, and you have the good sign everywhere, and you get a good estimate. If one of them is bigger than 1, but when the product is less than 1, you have to do it slightly otherwise, but basically it still works. So we are very happy because in some sense this was proving that the miracle which happened with the SKT model was in fact could be done again if you moved around a little the coefficients of the system. And as you know, in biology and population dynamics, it's not like in physics in which you have sometimes an equation with a very precise coefficients and you're sure that the coefficients are what they are. Here in biology or population dynamics, you only have access basically to order of magnitudes, and you cannot really assume that things are quadratic. When you put quadratic, it could be a different exponent. Anyway, that was in some sense good news that this structure could be maintained when you change a little the coefficients. And at this occasion, we were in some sense happy to be able to provide some kind of a general structure for the cross diffusion equation. I would like to try to explain it to you briefly. So this is actually a computation which is very close to the computation which is done for hyperbolic systems when you're looking for an entropy. And of course, we are also here looking for an entropy, so it's not such a surprise that there should be, let's say, analogies between the two. So let's say that you start with a system. Now, capital U is a vector, and you look at dtu minus Laplacian of R of U, and R here is a nonlinear function which goes from R to the I to R to the I. And all of this is equal to some reaction term that I will immediately abandon because it does not really play a role in the computation. So let's suppose that R is equal to 0, and you just focus on the cross diffusion term, which is A of U. Let's take some function which goes from Ri to R and call it phi, and you look at the derivative of phi of U. So then when you compute the derivative in time of this, you will get basically a gradient phi of U times dtu. But dtu is nothing but Laplacian of A of U. So you end up with what is written in the first equality in the slide. That is the scalar product in Ri of gradient phi of U, Laplacian of A of U. And then you do an integration by part using the Laplacian that you have. So when you do that, it may be better to write it in coordinates because it's a little easier to follow. So then you can write that this is minus the sum over all the coordinates of a big matrix, which is this Hessian of phi of u times the Jacobian matrix of A of u, which is considered as a quadratic form acting on the derivatives of u. And another way, so if you want it to now to be non-positive, then typically the good condition on phi is that somehow the Hessian of phi of u, which is a matrix, times the Jacobian of A of u, which is also a matrix, you take its symmetric part, and this is now a symmetric matrix, and it has to be positive in the sense of symmetric matrices. So that is a natural condition for phi to be an entropy. And it's vaguely reminiscent of what you do when you have hyperbolic systems. And so somehow this is the general theory, but as you can see, this is a computation of 3 lines, so it's not like it's a big thing. What is interesting is that there is something happening when this capital A is a function of 2 variables. So when you have systems of 2 equations, and those systems write like x1 times a function of x2 and x2 times a function of x1. And this is exactly the situation of the SKT system and of the generalizations of the SKT system.
Excuse me, can I interrupt?
Yeah, please.
Just a question. When the structure is that you have the Laplacian of something, would it work if you have instead like a chemotaxics?
No, unfortunately, I can say immediately no. And this structure is really related to the fact that you have the derivatives, the second derivative of something, and not divergent of something times the gradient. Okay. So there is really a different structure when you have let's say chemotaxis type structure. And then, of course, one can still try to do things, but unfortunately not this one.
Okay, thanks.
And yeah, so the point is that for the spatial, let's say, structure of capital A, then it's quite easy to build an entropy by the following recipe. So let's suppose that A1 and A2 are increasing. That is the case in the SKT system because it can be either linear or affine in the original system or some positive power in the generalized system. And suppose also that the determinant of the Jacobian of A is positive, which can be rewritten by this inequality, which is on the slide. Well, which has to be checked for all x1 and x2 positive. Then the recipe goes like this. You take these quantities AI, you take their derivatives, you divide by z, so you take a prime j of z divided by z, and you take now the second primitive of this quantity, you call it small phi I, and then the entropy will be the sum of phi 1 of x1 plus phi 2 of x2. So this is a recipe, and it's not very hard to see that it works, because for the form of capital A that I have chosen, it's quite easy to compute the Jacobian matrix. It's written here on the slide, and it's also using the proposed. It's also easy to compute the Hessian of phi, and it is actually a diagonal matrix with the entries a prime 2 of x1 divided by x1, and the same for x2 in the other part of the diagonal matrix. So when you multiply those two matrices, it's still something which is quite simple algebraically, and you end up with something which is written on the slide, and you can immediately see that you get a symmetric matrix. So you don't have to take its symmetrized part because it's already symmetric, and you just have to check whether or not it is a positive matrix. And now, since you have supposed that A1 and A2 are increasing, it's obvious that the trace is positive, and you just have to check if the determinant is positive. But the determinant is the product of the determinant of the Hessian of phi and of the Jacobian of A. And for the Hessian of phi, it's immediate to see that it's positive. And so you end up with the condition that the determinant of the Jacobian of A is positive, which was the assumption in this computation. Okay, so to remain simple, let's just recall that you have a recipe which gives you a Lyapunov-like functional. Actually, when there is no reaction term, it's a real Lyapunov functional for the system when it has the right form. So we are quite happy with that, but it does not answer the questions in many cases. First, when capital A is not of this form, and actually there are models which are interesting in which it is not of this form, and also when there are more than two equations. It's quite easy to see that when you have three equations, there are cases in which you can still produce an entropy, and I give on this slide one example. So here you suppose that you have some S which is not too large, and you have some kind of mixture of cross-diffusion terms between the three species. It's a model which makes some sense if you have the three species which are interacting by competition, but it cannot be found in biology books anyway. And the point is that for this model, it's still possible to find an entropy. And up to now, all the entropies that I have presented have this structure that it's a sum of functions of each of the components in the equation. So it's a phi1 of u1 plus phi2 of u2 plus phi3 of u3 here. And I have presented up to now only entropies of this kind. So it's quite natural to wonder if there would be cases, maybe for capital A different with a different structure, if you would get entropies which are different. And so actually, by discussing with various people, I discovered that there is a model which belongs to the folklore of people looking at cross diffusion, which writes like this. So this is actually very close to the SKT model, but you put inside a dependence of the cross diffusion, which is with a square. So you have Laplacian of uv square and Laplacian of vu square. So for this very special model, it's actually possible to write down an entropy, which is just the integral of u square v square. So as you can see here, you have an entropy which has not at all the structure function of u plus function of v. It's in some sense, it's completely dual. You have a function of u times v. And if you compute let's say the dissipation of this entropy, you end up with this trench formula in which you have gradient of u over u and gradient of v over v, which are appearing. They are appearing at two different points, and one can wonder if it's possible to use all of this in order to produce weak solutions for the system. And actually, I've been working on this problem with Ayman Moussa for some time now, and our feeling is now that actually it's not possible. And now our conjecture is that there is blow up for this model. So we don't know yet how to prove. Our feeling is that the entropy structure is not sufficient to provide a good theory for weak solutions of this model. So all of this is quite mysterious, and in some sense, we don't know how much energy we have to put in this, because of course the model which is here is a model that is invented in some sense. And it's not a model which is coming out of biology, and so we don't really know how much it is interesting to spend time on it. Anyway, just to show you that there is no reason to restrict oneself to Lyapunov functionals, which should be sums of functions of a given variable. And so together with Ansgar Jungel and Esther Daos, we actually thought that there is yet another way of producing entropies, which is quite different and which is based on the idea that those cross-diffusion systems sometimes are limits of fast standard reaction diffusion systems. By fast, we mean that the reaction which appears in the right-hand side is fast in the sense that you would put 1 over epsilon in front of it, and you will try to let epsilon go to 0. So a typical example is written on the slide, which I am showing now, in which you have on the right-hand side of the equation for U, V, and W. So you imagine here that U, V, and W are, let's say, concentrations in of chemical species. And you imagine that you have the reaction V plus W gives U. Then the mass action law tells you that the right-hand side should be like VW minus U, VW minus U, and sorry, minus VW minus U and minus VW minus U, because of course each time V and plus W gives U, you get 1 U and you lose 1 V and 1 W. Okay. And now you put 1 over epsilon in front of all those terms, which means more or less that the time on which you are looking at the chemical species is very large in front of the typical time of the reaction you're considering. It's already an interesting problem of ODEs, which can be solved by using, let's say, a singular perturbation theory. But it becomes really extremely interesting once you add the spatial structure. And so now you suppose that you have a diffusion for U, V, and W. So you could put a standard diffusion with coefficients like D1 Laplacian U, D2 Laplacian V, and D3 Laplacian W. But it's also interesting to look at it when you put a more complicated diffusion, like nonlinear diffusion of quadratic type, which is written here. Because what I will do now will be something which does not really depend on the kind of diffusion that you put. So the idea now is that the standard chemistry theorems tell you that for when you have a situation in which you have a reversible reaction like this, then you have an entropy which is given by, once again, U log U, V log V, and W log W. So you add all of this, and you know that it is an entropy of the system for any epsilon. And now you can pass to the limit when epsilon go to 0. So if you try to do it rigorously, it's not easy. And actually, for this special model, it was proven by Bote and Roland a little more than 5 years ago, I think. But here, it's not really my point. My point is that you can do it formally. And when you do that, well, you can see that you had 1 over epsilon times VW minus U. So in the limit, you expect that U is equal to VW. And also, you can add the first and the second equation, and this kills the right-hand side. And you can also add the third and the third equation, and it also kills the right-hand side. So when you do that, you end up with the macroscopic system, if you wish, which is a limit when epsilon goes to 0 of the fast reaction equation. This is a system which is a little complicated because it is somehow implicit, but you could make it explicit by defining a new variable, which would be VW plus V, and another one, which would be VW plus W. And here it's not very difficult because all is quadratic, so you could really compute explicitly things and you would end up with square roots everywhere. And the point is that you have now a system which is a cross-diffusion system, and the entropy that you had for any epsilon is of course still working when epsilon goes to 0, and so you have naturally an entropy without, I mean, it's given to you by the chemistry in some sense, And what is fascinating from my point of view is that now that you have done this formal limit in which at the end u is equal to VW, then you naturally have a structure which is not a sum of a function of V and a function of W. So now you have naturally a function of V plus a function of W plus a function of VW. And so this shows that there are systems which actually have something to do with physics, even though they are not maybe the most interesting ones, in which you have entropies which are not of this special form. And so the recipe that I proposed at the very beginning, it does not, in some sense, it does not give you a complete view of the kind of Lyapunov functional you can be expecting. And this is a typical example. But once again, you could tell me, well, this is a very special case, and it gives you finally an entropy when you have a very particular algebraic structure, and this may be not at all, let's say, it might not be generic at all. And so together with Ansgar and Esther, actually, we provided the following theorem. So here it's not written in a very rigorous form, but it can be made completely rigorous. That in fact, when you have this entropy structure, in some sense, the set of equations in which you still have an entropy structure of the same type is an open set. So this means that if you take a case in which you have an entropy structure which is obtained by a fast reaction limit, then by adding a small amount of extra cross diffusion in the Laplacian for u and v, then you still get an entropy at the end. So this is written here in a precise way. So you add now eta up of u and v in the first equation and eta v of times q of u and v in the second one. You suppose that you have a certain number of restrictions on the signs of the p and its derivatives. And when eta is small enough, then you still have this entropy which existed at the level of epsilon, which is still an entropy for the final equation, which is written here. So it's the same entropy as previously, and it still works for an equation which is slightly perturbed with respect to the equation that we had previously. Well, of course, this is just an example, but this means somehow that this entropy structure, it is in some sense structurally stable, which I think is quite interesting because it means that you don't have to have a very special algebra to get such a structure, and it gives some hint that hopefully it's possible to get weak solutions for cross diffusion equations for a set of equations which is not of measure 0 in the set of all possible equations, if this has a sense. I would like to finish by just saying a few words on the last thing that I did on the on this question of the entropy structure for non-triangular cross diffusion equations. So actually, you remember that Chen and Jungel proved the first entropy structure for a cross diffusion equation, and actually they worked again, but in fact it's not the same Chen, it's a different Chen. And together with Esther Daos, they actually showed that it does not work only for a system of 2 equations, but actually it works for a system of as many equations as you wish, provided that you have some link between the coefficients which are now called AIG and which are the cross diffusion related to UI and UJ. So this is what they call the detailed balance condition, and it means that somehow if you have some set of constants Pi which satisfy Pi Aij equal to Pj Aiji, then you can have an entropy structure. and it has the same form as the original SKT, and you have to put in front of the logarithmic entropy, you have to put this pi in front. So if you think of it a little, it makes sense, because now the Aij represent, you have a set of n square coefficients if you have n equations, and for the entropy you have the possibility to put only n parameters in front of the different entropies. So for it to work in some sense, you need to have a condition which sort of projects the n square parameters on only n parameters, and this is the detailed balance condition. So it's a very nice result, and actually I will not say more than this now, but the point is that together with Esther Daos and Helge Dietert, we were able to provide actually some microscopic model, which is a Markov chain, which is reversible, and for which therefore there is an entropy at the discrete level, which in the formal limit gives you the cross diffusion equation, the SKT system for an arbitrary number of equations, provided that you have the detailed balance condition. And so in some sense, I will not detail it at all, but let's say that in some sense we are able to explain the existence of this entropy because we have an underlying Markov chain which is known to have an entropy by standard theorems for Markov chains. So maybe this is all I wanted to say on the non-triangular case. So as you can see, we are very far from having understood everything, but we have at least a few cases in which we know where from comes this entropy structure, and we know how to build an entropy structure in some cases. And we also know that when we have found an entropy structure, it still works if you modify a little the parameters of the equation. That is more or less the state of the theory. Of course, our hope is that we can know more, and maybe at some point, if you give me a cross diffusion system, maybe of two equations, I will be able to give you the entropy. That would be the ultimate goal, but we are still very far from it. I just want to finish this talk by saying one word on the non-triangular case, on the triangular case, sorry. So this is a case in which one of the cross diffusion is not apparent in the equation. And actually this case appears in many problems, especially in problems in which you have some Turing instability at the end. And so we have started a collaboration with Lucilla Corrias, Elisabetta Brocchieri, and Helge Dietert on this theme. And now we also have a lot of partial results, let's say, for entropies in this special case, which is in some sense quite different from what I have showed to you today. And so this is, I would say, now our new line of research in this for elucidating the structure of the cross diffusion equations. I propose to stop here.
Thanks to Professor Desvillettes. Any questions, comments?
Well, just as a matter of curiosity, I mean, there is someone used the Rényi entropy in conjunction with a nonlinear scalar heat equation to derive some sharp form for the Gallardo-Nirenberg inequality.
Yeah, sure.
Do you see anything similar possible like in this context?
I could imagine, let's say, but yes, there is all this group with Carrillo, Dolbo, etc., who are working on the link between inequalities and, let's say, PDEs in general. So it can be, as you said, let's say, nonlinear heat equations. It can also be like Fokker-Planck type equations, and they usually give different kind of inequalities. And so one can imagine that there is a link between some inequality and the cross diffusion equations. I've never seen that, but I agree that it's it's a natural thing to try to understand. Yeah.
I have a small question. Most of the entropy you showed are the classical type of Gibbs-Boltzmann entropy, and so we know what is the physical meaning. But what is the physical meaning of the Remy entropy?
Yeah, that's my point is the following, that when you start from a system for chemistry in which you have chemical reactions, then you know that you have the Boltzmann-Gibbs entropy when it's reversible, and you end up with things which are directly related to that. So it can be sums of those quantities, but when you do fast reaction, as we saw, you can also have more complicated things, but which still are related to these U log U type entropies. When you go to biology or population dynamics, my feeling is that for the SKT system, it's a sort of coincidence that the first entropy which was obtained is also of Boltzmann-Gibbs type. And my feeling is that it was found because those people had in mind the Boltzmann-Gibbs entropy, and of course they tried it and they saw that it worked, and so that was perfect. But my point is that it's most probably a coincidence. And all the other entropies that you can find when you modify somehow the equations, my point is that they don't have any physical meaning. It's just it just helps you to understand the structure of the equation, but I wouldn't give any kind of rationale of those things.
Hey, thanks.
Thanks again, Professor Desvillettes.
Can I ask one more question?
Oh, yes, of course.
Yeah, I mean, you know that, well, maybe it's a very naive question. You know that the Shigesawa Kawasaki Teramoto developed Turing patterns. Is that a contradiction with this existence of this monotonous behavior solution?
No, not at all, because the point is this becomes a real entropy in the sense of a Lyapunov functional, which really decreases only when you remove the right-hand side, only when you remove the reaction part. And then, of course, you don't have any Turing instability anymore.
Sure.
When you have the reaction part, it's not a Lyapunov functional in this sense. It's just something in which you have one part which decreases and one part which is bounded, which is very good for the estimates, but of course it does not tell you anything on the limiting behavior when time goes to infinity.
Okay, thanks.
Okay, I think there are other questions. No. Okay, thanks again, Professor Desvillettes, for the interesting talk. We stop here this webinar, and we'll see next week on Friday 9 with the 2 talks. We'll have Professor Luigi Preziosi from Politecnico of Torino and Professor Cinzia Soresina from University of Graz. I think that we may finish. Thanks again.
Thanks a lot.
Thanks, bye-bye.
Thank you, everybody. Goodbye.
Bye-bye.
Bye.
See you next week.
Bye.
Bye-bye.